Ladies and gentlemen, please welcome Cleveland Clinic's CEO and President, Dr. Toby Cosgrove, and Dr. J. Craig Venter, founder and president of the J. Craig Venter Institute.
Well, good afternoon and welcome to our Ideas for Tomorrow. Today is a very special day for us, and I'm delighted that you could be here with us. It really is my honor to introduce one of the most innovative and productive scientists of our time. His lifetime accomplishments and breakthroughs have been nothing short of spectacular. Let me just review with you in a short video some of those accomplishments.
J. Craig Venter is one of the leading scientists of the 21st century. A visionary, entrepreneur, and adventurer. His disruptive technologies have revolutionized science and changed the way we interact with the world. Craig Venter grew up in California. He rebelled against the rote classroom learning of the 50s, left high school, and spent a year on the beach, surfing by day and stocking shelves at Sears by night. Drafted into the Vietnam War, his interest in science began as a medic in the Navy hospital in Da Nang. Americans were taking heavy casualties. At 21, he was in the triage unit, where they decide who will live and who will die. Motivated and inspired, Venter went to college, earning a bachelor's degree in biochemistry and a PhD in physiology and pharmacology, both from the University of California, both in just 3 years. Dr. Venter's string of genetic breakthroughs began at the NIH and continued at his own nonprofit Institute for Genomic Research and privately funded Sorcerer Genomics. His innovative approach accelerated the pace of discovery. In 1995, he and his team decoded the first genome of a living organism, a simple bacterium. Then, in 1998, in a dramatic race with the publicly funded Human Genome Project, Dr. Venter made a bold announcement that he and his company were going to sequence all 3 billion letters of the human genome in just 2 years. Using his maverick techniques, Venter's team came in at a fraction of the time and cost of the government project.
In 2001, he and his group, Sorcerer, went to the White House along with a group from the National Institutes of Health.
We are here to celebrate the completion of the first survey of the entire human genome. Without a doubt, this is the most important, most wondrous map ever produced by humankind.
In 2003, his next mission? To launch a monumental effort to harness the biomass of the sea and help solve the planet's energy crisis.
Sample 128 off the coast of Africa.
We were looking for ways to try new fields that we thought would have the most impact on our future, the future of humanity.
To further advance the science and understanding of genetics, Dr. Venter led the combination of 5 affiliated centers and institutes into the J. Craig Venter Institute. Then, in 2007, he published and placed online the most complete human genome ever produced, 6 billion bits of his own DNA.
We've taken all the sequence from a single human, Craig Venter. We've characterized not just the single base pair changes that differ, but all the variation. Having your genome in your hand, on a disk, in a database somewhere, will enable you to then go to the biological literature that we're accumulating in many labs around the world and essentially put together a profile of your disease risk.
Having complete genomes gives us the ability to begin to unravel these mysteries of human evolution, human biology, and even maybe predict the future.
Recently, scientists at the J. Craig Venter Institute have gone from reading DNA to writing it.
These techniques could be very powerful for taking us to the next stages of applying biology to solve some of the world's problems.
In the past year alone, they have announced 2 breakthroughs that may enhance our power over nature beyond anything since the splitting of the atom. The first, a transplantation of the full genome of one bacterium into another. In effect, the first man-made species change. The second is the first synthetic replication of an entire bacterial genome, the longest DNA molecule ever synthesized, made with widely available chemicals, turning genomics into technology that may create solutions to the energy crises and global warming.
To engineer cells to produce biofuels, starting with carbon dioxide, and we've made a lot of progress down that road.
These breakthroughs move Dr. Venter and his team even closer to creating the first artificial organism, and one step away from the creation of life itself.
Recently, Dr. Venter completed his book, Decoding the Human Life. And I'd like to read to you the last few sentences of that book. I want to take us far from shore into unknown waters, to a phase of evolution, to a day when one DNA-based species can sit down at a computer to design another. I plan to show that we understand the software of life by creating true artificial life. And in this way, I want to discover whether a life decoded is truly a life understood. Ladies and gentlemen, please help me in welcoming Dr. J. Craig Venter.
Thank you very much.
Well, thank you, Toby, for the overwhelming introduction. It makes me feel my lecture has a difficult chance of living up to the introduction. But it's been an exciting ride for the last 20 years, and I think it's just starting to get more exciting. And I'm absolutely delighted to be here this afternoon at the Cleveland Clinic, a place where the advances in medicine are truly translated to patients, and maybe some of the work we're doing will help advance that even faster. You saw in the video a couple different views of my institute. We have a little over 450 scientists and staff, both in Rockville in Maryland and now in La Jolla, California, where we're expanding out there, as well as about 100 people in the synthetic genomic company, trying to make use and understand all this information we've been generating now for a while. I call the area that we've been through as the era of trying to digitize biology or digitize life. This really started on a large scale for us in the late 80s, early 90s, with the EST method in this paper in Science in 1991, where we went from a time where it took me 10 years to get one gene to this paper, which seems small by today's standards, of 337 new genes in one study. Now, this scaled up very rapidly, and we had to develop new mathematical tools to go further. Granger Sutton at then the institute was called Tiger, developed a new algorithm that allowed us to put sequences together in the computer very quickly. And we used this as an experiment to sequence the first genome in history. We didn't set out actually to sequence the first genome. We set out just to do an experiment, and it ended up being the first genome. The yeast genome project and the E. coli genome project, yeast took 10 years and 1,000 scientists. E. coli took over 13 years. Yeast ended up being the fourth genome finished. We reduced that time to 4 months with this initial project and a handful of scientists. And that changed even more rapidly. And it was only 5 years later when we scaled up to do the first version, the first drafts of the human genome. The public effort published at the same time in the journal Nature. But it turns out both these versions were wrong for different reasons. But we all thought in 1999 to 2001 that all humans had the same genes, and that we all varied from each other in only 1 out of 1,000 letters of the genetic code. Just last fall, we published the first now complete version of the human genome, the diploid genome, with the sets of chromosomes, in my case, from both my parents. So it's 6 billion base pairs instead of 3. And it was initially thought we could just infer the other half from having half of it. But what was missed was all the genetic variation and diversity. So now we know that we differ from each other by 1 to 3%, not 1 out of 1,000. And to put this number in context, up until this study, we thought we differed from chimpanzees by only 1.27%. So if that number didn't change as well, I'm hoping you're asking that to yourselves. We have some explaining about evolutionary biology to do. But that number has now changed to 5 to 6%. So we're not all just clones of each other. There is a lot of diversity, and that diversity creates a number of challenges for medicine, but it also helps explain a number of things in the medical community, and I think knowing the extent of diversity will help us going forward. Almost half my genes had 1 or more major variations in it, not just the single nucleotide polymorphisms, but insertions and deletions. In fact, we have more base pairs in insertions and deletions than we do in what everybody thought was the extent of variation of single nucleotide polymorphisms. But this extent of protein variation is very critical even in studying simple things like drug metabolism. You've all heard a variety of studies arguing whether caffeine is good or bad for you. Well, it turns out there was a good reason these studies were confusing. You can't do a study on a general population and get an answer. Depending on your gene set, caffeine can be bad for you, or it can have little effect. It turns out I have 2 variants for very fast metabolism of caffeine and many drugs. And studies have shown when you break out the fast metabolizers out of a group, that you can study the effect of caffeine on people who metabolize caffeine much more slowly. They have tremendous impact of caffeine on heart arrhythmias and increased incidence of heart attacks, while those like myself that can metabolize very rapidly, I can drink a quadruple latte a couple times a day and have no impact whatsoever. These same metabolism systems affect things like warfarin. So imagine trying to get the dose right for a patient, and you have 2 patients, one's a fast metabolizer and one's a slow metabolizer. If you use standard dosing practices, one would be overdosed dramatically and one would be not even reaching therapeutic levels. So starting to understand the extent of human variation will have an early impact on medicine. We're not just a single species as much as we like to think we are. We, in fact, have a large number of organisms associated with us, and we're at the earliest stages of understanding what they do. The techniques that we use for the human genome, we extended to the environment, and the environment can include the skin or various body cavities. And what we find is literally thousands of species. So if you're thinking of kissing the person next to you during this dark lecture hall, Think of the 1,000 organisms at least that they have in their mouth right now. And that might have you pay more attention to the lecture. We have only 23,000 genes in 100 trillion different cells. We have organisms with us totaling on the order of 10 million different genes. These vary tremendously depending on the environment we live in. Sometimes these organisms break through and cause disease. Other times they just sort of hang out with our bodies. When we look at the chemistry of humans, something that's hardly been studied at all, our 23,000 genes code for proteins that can make roughly 2,400 unique chemicals. Now, if we take a sample from your blood after a meal, we find around 500 major chemicals. Only 60% of those come from our own human metabolism. Another 30% come from the various chemicals extracted from what you just ate. But an amazing 10%, sometimes more, sometimes less, come from metabolites from the bacteria in our guts and our systems based on what we feed them. So this notion you are just what you eat is not quite correct. You're also what we feed our own bacteria. So imagine all these chemicals circulating in the blood. Pharmacologists and physicians like to think the only chemical that's circulating is the one drug that they're given to their patients. But in fact, that's on a background sometimes of hundreds to thousands of different chemicals. And that's a very different view of human biology than we've had before. So the environment is important. We're also looking at the environment on a broader scale, as you saw in the video, taking samples around the globe. As with our physiology, the ocean is packed with microbes. 1 million bacteria per milliliter, 10 million viruses per milliliter. I surf whenever I get the chance, and occasionally I swallow a mouthful of seawater, and now I stop to think about how many organisms I just consumed. Early last year, we published a special issue of PLOS Biology describing the first part of the Sorcerer expedition, where we took samples every 200 miles around the globe. This is the route that we followed, starting in Halifax, going down the eastern seaboard, through the Caribbean Sea, through the Panama Canal, and down to the Galapagos. The early set of samples that was described in this special issue, we had over 6 million new genes in literally hundreds of thousands of new species that were discovered. The 6 million new genes more than doubled the number of genes from all sources in the public databases. Our experiments were remarkably simple. We just filtered seawater, captured different size species on different size filters, took them back to our lab in Maryland, where we isolated the DNA from the filters all together at once, and then used the computers to sort that out. This technology is also changing rapidly. There's new techniques that don't even involve cloning the DNA, providing a much more rapid throughput, and this is lowering the cost, changing what we can do in the environment, and changing what we can do with humans. But what we found amazingly was every 200 miles, 85% of the sequences were unique. The ocean is not a giant homogeneous mixture. In fact, we can tell where a sample came from in the world simply by looking at the DNA in the water. Think of this with the ships that come into the port here and dump out ballast water, literally dumping out trillions of organisms while they fill up with cargo, go someplace else, and exchange organisms around the world. It's truly amazing. There haven't been more ecological disasters, but certainly the Great Lakes have suffered some of those. Now, the oceans are certainly parts of it. We're supposed to be deserts. The Sargasso Sea, where we started, is known as an ocean desert. So we were told by biologists we would find very little life there. In fact, we found just in a barrel of seawater 40,000 new species. And so how could they live? It turns out they have unique molecules almost identical to our visual pigments. They have photoreceptors. The blue at the bottom of this graph was our knowledge of photoreceptors prior to this expedition. The red were all the new ones discovered just in the first shot. And these photoreceptors capture energy from the sunlight and turn it into chemical energy in the cell. We literally have thousands of these now, and we can line these up and find, in fact, there's a single amino acid residue that determines the wavelength of light that these receptors see. So we can ask unique questions, because each one of our sequences is tagged with a GPS coordinate. So we can ask, is there a unique association with these in the world? In fact, we found, surprisingly, there was. In the middle of the ocean, the water's a deep indigo blue. The photoreceptors on the organisms that live there see primarily blue light. You get into coastal waters, there's a lot of chlorophyll. They see primarily green light. You go to places like the Panama Canal, where it's entirely fresh water, they see entirely green light. So it's only a single amino acid residue that determines this survivability advantage in the ocean, and a single base pair change in the genetic code can change that single amino acid. In fact, a Swedish group last year published a paper showing that these photoreceptors actually do provide the energy and drive the biology of these cells. This is not photosynthesis. This is totally an independent mechanism. These are the organisms that do a lot of the CO2 fixation, and it was an unknown process. We thought these were very rare molecules. It turns out to be one of the largest gene families on the planet. The other surprise is people measured diversity by measuring 16 sRNA, just one gene in organisms. In fact, what we found is that underrepresented diversity by orders of magnitude. SARA11 was supposed to be a single species, and a relatively rare one at that. In fact, each of these little lines that you see represents roughly 900 base pairs of sequence. We see a huge diversity range from 97 down to 60% identity, but the same genes largely in the same order. This is not what people predicted by Darwinian evolution of survival of the fittest. We have in parallel survival of thousands of related organisms. Some people are using the term pan-species to cover this group of related organisms, all with a similar environmental niches. We can take a slice of this data in any fashion and build trees out of it, and you can see there's tremendous diversity, but we can ask unique questions. Does it differ by site? These are color-coded. Is the Atlantic Ocean different from the Pacific versus the Indian Ocean? But the bottom is the most interesting to me. In recent evolutionary history, this switch between blue and green light for the photoreceptors has happened 4 times. So this makes sense. You get a random mutation if you switch to blue light and you're in the middle of the Sargasso Sea. You will have a tremendous advantage in survival over your colleagues that can only see green light, and vice versa into coastal waters. So this is much more like classic Darwinian evolution of you get a mutation, you get a tremendous survival advantage. So we see classic Darwinian processes, and we see novel ones that were unexpected. But you can ask any question of this type of data, whether it's from the ocean or from the human gut. A lot of additional questions. How much novelty is really added by this data? Are we really discovering new genes? Or like the photoreceptors, are these just members of known gene families that hadn't been seen before? And if we're making new discoveries, what's the pace of discovery? Some of you might remember in 1969, the US Surgeon General said, we've won the war against microbes because there was a new set of antibiotics. And microbiology groups, departments, companies began to shut down around the world. I was told in 1971 at the start of my thesis work in San Diego that it was going to be hard to make any new discoveries in biology because it was essentially all known. It seems so incredibly absurd today, but our thinking goes through bottlenecks, and this has happened at various times in science, and this certainly affects thinking from time to time. Fortunately, the bottlenecks are usually followed by periods of massive discovery. To try and answer some of these questions, we assembled all our data together with all the data we could extract from public databases. It took over 1 million CPU hours, and we came up with around 50,000 major gene families, those with at least 10 or more members in the databases. But there's a very long tail on this data with literally 1 million or more genes that look like they could be unique gene families. Keep in mind, we have 23,000 genes, maybe only around 16,000 gene families. So we represent a small part of the biological niche of genes on this planet. In fact, if we look just at the mammalian genes and look at the pace of discovery, we find that it's basically saturated. So if your goal is to discover new genes, you should not start with mammals or humans. We'll understand new variations, obviously, from each new human we do. If you find another mammal, you'll find unique gene content and gene order, but we know the gene set of mammals now. In contrast to bacteria, archaea, viruses, we're very much in an earlier linear stage of discovery. We can ask bigger questions of this data, and we've been doing that for a while. Things like, what is life? I'm sure most people have asked themselves that question at least once or more in their lifetime. Science has obviously proceeded by trying to pare life down to its basic components, this reductionist approach. We've answered the next question, can we digitize it? That's what we've been doing when we take this analog world of biology, sequence the genetic code, and put that code in the computer. So now the question that we're trying to answer is, can we regenerate life from that digital code? Ever since 1995, we've been working on trying to design and synthesize life. This was based on some early experiments with the first 2 genomes, looking at the gene content, the gene number, and how it differed, and asking whether we could come up with a minimal genetic code from an organism. We worked out the only way to answer these questions was to make a synthetic chromosome. But there's really tough technical hurdles with this, and we had the question, can the chemistry even permit making large DNA molecules or entire chromosomes? And it's taken us a long time to answer that question. And the second question is even harder. Can we, if we have this chemically made chromosome, an inert molecule, can we boot it up in a cell? When we first started this process, we found that DNA synthesis was a very degenerate process. It's error-prone. The longer you make the molecules, the more errors they have. So my colleagues Ham Smith and Clyde Hutchinson came up with some new methodology that allowed us to make molecules very accurately, quite rapidly. We actually started with a virus that kills bacteria, Phi-X174. It was the first true genome that was sequenced by Fred Sanger and colleagues in the late 70s. It's only about 5,000 letters of genetic code. But one of the key tenets that we're developing with this digital biology field is the process can only be as good as the digital data is accurate. We went back and resequenced the Phi-X genome and found only 3 differences ever since that first genome. So that was done with a much higher standard than a lot of data sense. And it took us 2 weeks from designing the pieces that we were putting together to build the molecule. And we had now this sequenced piece of DNA that we knew to be 100% accurate. We injected that into E. coli, and we're very pleased and surprised when E. coli started reading this synthetic piece of DNA and manufacturing the virus. As with many biological processes, the progeny was not very grateful. It turned around and started killing the cells that made it. And that's how you can detect it. These clear plaques that show up on this lawn of bacteria allowed us to isolate the virus. So we call this a case where the software is building its own hardware. The software we made chemically in a lab from the digital information, put that in a cell. The cell machinery read it and started building the viral particle. Our goal was not to build a virus, it was to build a bacterial chromosome to answer some of the questions that we were working on. And we thought we could build this in cassettes of viral-sized pieces, pieces on the order of 5 to 7 kilobases. And the question was, how would we put all these together? Well, first we had to go back to the design phase, and we were starting with Mycoplasma genitalium genome. It was the second genome that was done in 1995. It has a little over 500 genes. But again, we went back to resequence the genome. The standard in 1995 was 1 error per 10,000 base pairs. And we found 30 errors in the genetic code on resequencing it, errors that in fact would have permitted it from being biologically active. Again, proving this notion that the quality of the digital information is critical. Now think of the design phase. You can only make pieces 80 letters long. You have to design these so they overlap each other to go together to produce pieces 5,000 to 7,000 letters long. They have to be designed so they overlap with the next pieces and so on. And the thing we were worried about the most was a contaminant, an artifact. One single molecule of the native chromosome from a cell could mislead all of us thinking we had something when we didn't. So we developed the notion of watermarking the genetic code, and this has turned out to be a fun phase, and a lot of people have gotten excited by this. We have the 4-letter genetic code, A, C, G, and T, and we know that triplets of that code for amino acids, and there's roughly 20 of those, and there's a single-letter designation. So we can use that to use the genetic code to write words, letters, even sentences. People were upset that we weren't more profound with what we put in the first genome. We basically put in the authors' names of people building the genetic codes who we were autographing it. Our main method and reason for doing it, as I said, was to make sure we could distinguish the man-made chromosome from the naturally occurring one. So the process we started on was to put these pieces together. We would clone them in E. coli, sequence them to make sure that they were accurate at each stage. But as we got to bigger and bigger pieces, and the largest piece of DNA that ever had been made before was only 30 kilobases. And so pretty soon we were in new territory that people had not been in before. And with the larger pieces over 100 kb, they didn't grow well in E. coli. And so we started growing them in yeast and made an important discovery. We had been studying Deinococcus radiodurans as a means of assembling the genetic code. This is an organism that can take 3 million rads of radiation and not be killed. We recommend you not try that at home. Humans can't take so much radiation. In fact, we can only stand a tiny fraction of that. The top panel here shows what happens after 1.75 million rads. The chromosome gets blown apart into small pieces. But 12 to 24 hours later, it stitches its chromosome back together exactly as it was before, and it can start replicating again. So this is an organism that we now know can survive in outer space. It can be in a vacuum. It can take a lot of ionizing radiation. And we have thousands of organisms like that on this planet. So it won't surprise me soon if we discover similar organisms on Mars. We exchange roughly 200 kilograms of material between Earth and Mars each year, so I'm sure we're exchanging genetic material as well. We thought we could use these repair mechanisms to assemble genomes and had been working on it for some time when we found that yeast just does this naturally. So as we put these large pieces in yeast with designed overlaps, yeast just assembled them nicely and built the entire chromosome for us in a very short period of time. This is an actual photo of the piece of DNA. It's such a large molecule, we can see it with a light microscope. We don't need an electron microscope. It's a circular chromosome. It's over 300 million molecular weight, making it the largest human-made chemical of a defined structure by a substantial margin. It's over 580,000 molecular weight, or base pairs. So if we printed these out with no spacing at 10 font, it would take 147 pages to print out this genetic code that was entirely accurate. The big challenge now is trying to boot that up in a cell. And this is something we've been working on in parallel, and last year published what I consider the key study on doing this, where we actually transform one species into another by changing its life software. We did this by isolating a chromosome from one mycoplasma species, in Mycoides. We treated this with digestive enzymes to get down to naked DNA because we wanted to prove that you didn't need any proteins along with this. Otherwise, the synthetic approach would be problematic. We added a few extra genes to it so it would turn cells blue and we could select for the molecule. And then we inserted that into a closely related species, roughly the same distance as humans and mice are to each other. And there were a few other aspects of this. I'm making a very long story short. We thought we would have to destroy the chromosome in the cell before we transplanted in the new one. When you think of cloning with eukaryotic cells, it's a pretty simple mechanical step. You just remove the nucleus from one cell and replace it with another. It's a pretty simple procedure. With bacteria and archaea, the chromosome is integrated into the cytoplasm of the cell, so there's no way to do that. So we chose cells with specific characteristics. The capricolum genome has no restriction enzymes, the enzymes that chew up DNA. And let me show you with a very sophisticated graphic what happens. We inserted the chromosome from the Mycoides genome into this capricolum cell. And this chromosome that we inserted coded for a restriction enzyme that was immediately expressed by the cell machinery in the cell. And that enzyme then recognized the capricolum chromosome as foreign DNA and chewed it up. Therefore, we were left just with cells with the transplanted chromosome. These cells became blue. All the characteristics of the initial species were gone within a few generations. All the proteins now in the cell were those coded for by the transplanted chromosome. So this was, I guess, the ultimate in identity theft. And it's why you can, my best friend and colleague, Ham Smith, received the Nobel Prize in 1978 for discovering restriction enzymes. I always appreciated them as tools in molecular biology, but I never truly, until we did these experiments, appreciated their role in evolution. Obviously, if a piece of foreign DNA could just come into you as a species and completely steal your identity, you would want some protection against that. It would certainly change the nature of sex, wouldn't it? Some people think that's what happens anyway, but cellular systems develop restriction enzymes to recognize the DNA as foreign and chew it up. But this doesn't happen in every case. In fact, in contrast to the types of evolution that I showed you with the photoreceptors, we find lots of cells with multiple chromosomes in them. Cholera, for example, when we did the cholera genome, it had 2 chromosomes, very different from each other. They either resulted from a cell fusion or from taking up DNA. The spectrum of restriction enzymes doesn't always work, and for a variety of reasons, you can collect more chromosomes. But instead of a point change where you change one letter of the genetic code and change a feature like the color of the photoreceptors, by chromosome transplants or cell fusions, you can change thousands of characteristics of the cell in an instant. And we see evidence of this happening over and over again in evolution. So in fact, what we were trying to do in synthetic biology told us a lot about what happens in nature. We have not yet done this with the synthetic chromosome. It's different than the Mycoides genome. And each system we have to optimize the restriction enzyme systems, the degree of methylation of the DNA, et cetera. So it's not a trivial process. but we hope to have the synthetic chromosome booted up later this year. So while we've been doing this process over the last decade or so, we've thought of a lot of important potential applications of it. And we're trying to think of this new field of synthetic genomics much like the electronics industry was in the 1950s. Only there we had only a few dozen or so components: resistors, transistors, capacitors. Now, right now in our database, we have 20 million genes. And we like to think of these as the design components of the future. Imagine with 20 million different variants what we might be able to come up with. So this is part of our design software for designing new species to do specific purposes. And the purposes, I will show you, are almost unlimited. And we have a new area we're developing we're calling Combinatorial Genomics, where instead of taking 4 years like it did to build that one molecule, we can now use robotic systems and things like the yeast cell to build the chromosomes very rapidly. So we're trying to get up to thousands to perhaps millions of chromosomes a day. With these 20 million genes we have in the databases, very few of those have actually been very thoroughly studied. So we need new combinatorial methods to try and work out what they do. And we have to use empirical processes in biology, screening for selection, whether it's for chemical production, just does that combination give you a living cell, antibiotic production, et cetera. And I think this is going to change things quite rapidly. We're trying to use these tools to come up with what we think would be some solutions to the biggest problem facing humanity. That's what we're doing to our environment, with adding right now 4.2 billion tons of CO2 a year to the atmosphere and increasing at a substantial rate, primarily from burning oil and coal. So we're trying to see if we can get regenerated carbon from a variety of processes and making much more advanced fuels than ethanol. We're not short of energy on this planet. 120,000 terawatts a day reach Earth. We only need to capture a tiny fraction of that to replace all the fossil fuels that we're using. Our ultimate fuel, we think, will be based on carbon dioxide. The problem we've seen with this experiment that's being done in this country of converting corn sugar into ethanol is not sustainable. And in fact, has shown the world what a silly experiment it's been, using food land to produce fuel at a time when our population is going to go from 6.5 billion to 9 billion people. And we don't have sufficient food or sufficient fuel. So we need independent feedstocks, CO2 being the ultimate one. Sunlight is a great way to fix CO2 into cells. That's what corn and other plants are doing. They just don't do it very efficiently. And also we have other mechanisms. The first archaea that we did in 1996 uses CO2 as its source of carbon. Molecular hydrogen is its energy source, works at high temperatures, and converts that CO2 into methane. We're working on a number of programs. BP is a partner to convert subsurface coal into methane. So you don't have to mine the coal, you just use biology to convert it. So we're looking at a variety of new sources: sunlight indirectly through plants, fixing carbon dioxide as a source of replacing the petrochemical industry, source of the chemistry for making pharmaceutics, bioenergy, et cetera. We've had some successes with this over the years, or they've been slow. In 1996, we started the first reverse vaccine experiment with what was then Chiron and is now Novartis. We're trying to come up with a new vaccine for meningitis B. It's taken 10 years, mostly due to FDA restrictions in doing clinical trials in children. But now there's a very effective vaccine based on genomics that works against almost every strain of meningitis B. You can see it here in contrast to existing vaccines, which are not very effective. We think synthetic biology can totally change how we do vaccines, because we can now design a bacterial cell that expresses every childhood vaccine molecule on the surface, maybe replacing them all simultaneously, and also using it to design vaccines for things that have been very resistant, such as malaria, by being able to express multiple variants on the surface of a cell. Bert Vogelstein's had some amazing effectiveness by using Clostridium as an anti-tumor agent. So Clostridium works in an anaerobic environment. When injected into patients with tumors, it homes right in on the anaerobic part of the tumor, releases substances that end up killing the tumor cells. So we're actually going to try and design some bacteria that do this that wouldn't lead to infection, but could be designed to release very specific toxins and have unique characterized properties. So we're just in the earliest stages of this new field as we go from reading the genetic code to trying to write it. And as usual, I'm optimistic that this can have an impact, hopefully for understanding the basic elements of life, what we started out to do the experiments for, but maybe to help us survive as a species, replacing the petrochemical industry, producing new energy, enhancing bioremediation, but also some of the experiments that I showed you with a new vaccine and anti-cancer, new therapeutics, combinatorial vaccine production. The first question we always get is the ethics of trying to design new life forms. We asked these ethical questions before we started the first experiment. A 1.5-year long review at the University of Pennsylvania was published in 1999 in Science. They brought in almost every major religion to consult them. The consensus was we were taking reasonable approaches and should proceed, the caveat being concerned about biowarfare. The Sloan Foundation funded my institute along with MIT to do a much more thorough review that was published this last fall. The US government in the executive branch has a new committee, the NSABB, that involves every branch of the federal government, and they approved the making of the 1918 flu virus at the CDC from DNA and RNA that was isolated from people that had died in 1918 from the flu. This has led to understanding the source of the most lethal parts of the virus and designing of new vaccines. So there are processes, it's discussed widely, but it's going to be an ongoing discussion. My last slide is just a key part of what we do at the Institute is also public education and science. We started a bus in the DC area that goes to middle schools. The studies in education have shown that particularly for women and minorities, you need to capture their imagination before they go into high school. And it's been very effective. This goes around to middle schools. The schools were initially skeptical. Now there's a 3-year waiting list for the bus to go there. The kids do things like CSI, crime scene solving, other things that I could only do in science long after I had a PhD. So the scale of what's being done is affecting people. The feedback on this has been tremendous. And I think there are a lot of ways to change the future as we go forward. Thank you very much.
Fabulous. Thank you, Chip.
Paris. Nice
to meet you.
Well, Craig, I can't thank you enough. What an absolutely stimulating lecture. And I knew that when I come to talk to you that I need to take an expert into the booth with me. So Dr. Karis Eng is head of our Genomics Institute, and she's going to join me for moral support and certainly intellectual. Craig, I had to ask you, and many people I think are concerned about this, as you are the only person really who's been completely decoded. And I must say that I thought a little bit differently about you when I realized you had a proclivity for alcoholism, coronary artery disease, obesity, Alzheimer's, and antisocial behavior.
Not to mention high-risk behavior.
And I wonder how that made you feel about you.
Well, it's been interesting. I've been most interested with the Canadians' fascination that I have watery earwax. So you have to be have a certain immunity to people looking too closely. But it's the earliest stages of interpreting the genome. And unfortunately, most of the genetic studies are looking under the proverbial lamppost for results. So if you look for some genes that are associated with diseases, you can find changes, but they don't necessarily prescribe your outcome. So as you probably saw, I have an increased risk for Alzheimer's disease.
Yes.
There's no history of Alzheimer's disease in my family that I can remember.
That you can remember.
And we know there's genes associated with longevity and health. I have some of those. At the same time, I have these risk alleles. And so having one genome, and soon to be two, in a week or so, Jim Watson's genome is going to be published in Nature. But we don't need one or two, we need tens of thousands.
Yes.
So we now know sort of the scope of the problem, but to get the answer, we need complete phenotypic information on individuals and complete genomes, and we need them on the tens of thousands. And if we have that, we'll be able to answer almost every question that any of us have had about nature versus nurture, but also in terms of risk, like a Framingham type study, only having the genetic code. It's been very unfortunate, in my view, with some commercial companies having tests for things like breast cancer. They push those tests as though those one or two genes will really tell people what's going to happen in the future. A lot of people have taken that too far and have had radical double mastectomies, think that will be a preventative measure, when the information was incomplete in the first case about what the risk levels were. So we need to look at the whole picture. What prevents disease? What leads to disease? And so we're at the earliest stages. So I don't take anything too seriously in the genome. Some of the things that people look at, the alleles, are more for fun than anything else, thinking that a single change in a dopamine receptor will determine your risk-taking proclivity. Maybe it does, but I think those would be real surprises for humanity if we're that simplistic of a genetic machinery.
Karis, breast cancer is right up your alley.
Therefore, I will not pursue that. So personalized medicine has grabbed the imagination of both the public, the scientists, and the physicians. Now, I've always said that ADO typing is, in fact, one of the earliest types of personalized medicine. Why has it taken off and just grabbed everybody's imagination right now?
Well, I think there's several new companies now pushing it. There's not been too much in science that captured the imagination as the race to sequence the genome. I don't think any of us realized at the time we're too caught up in the race. But even if people don't understand science, they certainly understand competition. And that was the introduction of genomic science to most people. And now we've had a lot of interesting results about some predictions about where things could go. And that's, I think, intrigued people. All this work of just using the mitochondrial genome to track your maternal ancestry, the Y chromosome to track paternal ancestry. I think there's a fascination with that. So these companies like 23andMe and Navigenics, Decode, that do an assay for $1,000, I think it's actually great because it introduces people to genomics and genomic tests, as long as they don't take the information too seriously or don't try and act on it other than seeking more information from their clinicians if there's something of question there. But I think it needs democratization. The NIH is going the opposite way. It's saying this information is so valuable it has to be kept secret, and you have to have a retinal scan to be able to look at the databases generated with public money. So I applaud Jim Watson for following me and putting his complete genomic information on the internet. And we have literally thousands of volunteers that want to do the same, which I think is what will move medicine forward.
We got time for just one or two questions. I met somebody, you want to bring the house lights up, and maybe that there's some questions that some of the audience would like to ask, and then we'll let you think about dinner. Yeah, let's start over here.
Hi, Dr. Venner. Can you hear me?
Yes.
Hi. I just wanted to say before I get to my question that as a young scientist, you are both a hero and an inspiration to me. And I thank you so much for coming to the Cleveland Clinic today and sharing your ideas with us.
Well, thank you for your nice comments.
My question for you is, as in your career you've started so many of your own institutes and foundations and companies to do research, and it seems that one of your reasons for doing this was to make research happen faster. You weren't satisfied with how fast research was happening in the scientific establishment. So my question for you is, what advice, suggestions do you have for changes that need to be made to the scientific establishment and the way we do science, so the rest of us can start making discoveries as fast as you are?
It's a great question. It's hard to give a short answer to it. I mean, I think part of the frustration that we've had is our government system, particularly through NIH and NSF, has a huge budget. But the government is, and the way we dole out money for research is the ultimate conservative way to do it. None of the discoveries that I showed you that we have made started with a funded grant. But after we showed the new methods work, the money came pouring out. So everything we did, we had to start with independent money. That was the motivation for starting the company. It was the only way to get money to do the research we were doing. So if you get turned down on a government grant, don't let that be the end of your career. Say thank you, there's better ways. And they are faster. But we need to try and change the system so it's more responsive. But I was on Zuhuni, the NIH Directors Committee, to give out an award for the most innovative high-risk science. And the people on the committee came up with a list of recommendations. And NIH did not give the award to one of them. They wanted to give it to safer people that weren't really taking risks. It's not in the genetic code of government people to take risk. And that's a fundamental flaw with how we do science funding. And we have to change it. Things like DARPA are able to take risks because that's what they're charged with. Maybe the government needs to give the money to an independent organization that can make much better peer review decisions. But it's a huge challenge, not just facing the US, but facing the world.
One more question over here, and then we'll...
Hi. I wanted to know if you thought we're close to curing anything in our lifetime, any disease you can possibly think of, or is there anything we're getting close to?
That's a question for Toby, right?
I'm a knuckle-dragging surgeon.
Well, I mean, there's some diseases that certainly in my lifetime have been eliminated. The vaccines are the most effective way to treat disease. When we look at the polio vaccine, for example, it's virtually 100% effective. Most pharmaceutics are about 30% effective. So I think the only way we're going to start truly eliminating disease is by going back to a much more effective vaccine program, and ultimately perhaps from changing the genetic code to eliminate some of the more devastating diseases from future populations. But I think that's a big risk for the future. Right now, if we can effectively treat diseases better, that's going to be a huge accomplishment. I think eliminating disease is something that's proven more of a challenge than the scientific community has the equipment for right now.
Well, Craig, on behalf of all of us here, I want to thank you for sharing your expertise with us today, for stimulating us, and for, more importantly, being a model for what can be accomplished by determination and hard work and the progress that you have made in taking us into a whole new era of science. And I cannot thank you enough, and I want to tell you that the next speaker will be Oprah Winfrey, who will be here on May 12th. Craig, thank you very much. I appreciate it.
Thank you.